I think I'll just start. The rest can join us whenever they'll find us wherever we are. Okay, so I'm going to take you through folk medicine. We all know folk medicine is also known as traditional medicine. It's what our parents, grandparents use, specific remedies that our parents have gotten from our grandparents and the other ancestors. So, so, okay, uh, I'll just take you through examples of camp. So there's the African traditional medicine. There's the Chinese, which involves the acupuncture, the herbology, food therapy, Twina, Tai Chi, and moxibustion. And okay, so moxibustion is basically using of incense sticks, where they just erect the place that you are in to enable the therapy. Ayurveda is what the Indians use. Unani is what Japanese use. Homeopathy is, well, it originated from Germany, but it's one that stimulates healing and strengthens the body's ability to heal itself. Then there's the naturopathy, where can you guys hear me?
Yeah.
Okay, I'm sorry the background noise is. My nieces are making noise. So you'll just forgive me for that. So naturopathy is simply alternative therapy, which is based on treating diseases by the use of drugs and techniques such as controlling your diet, exercises, and massage. Then osteopathy is basically manipulating your body structure, so your bones. It's what chiropractors do, but there's a sense of, it's basically what chiropractors do. Campo medicine is where you tend to enhance the motility of your gut and you promote your digestion. Orthes is spiritualism, diviners, and bone setters. So I don't know how different bone setters is from chiropractors, but I feel like it's the same. Then spiritualism and diviners is basically meditation, and those guys who get you in touch with your 7 chakras and all. So access to CAM, there is the historical and cultural beliefs, there's the socioeconomic status, inaccessible conventional health system, belief that natural is risk-free, and adverse effects of conventional chemical drugs like chronic diseases. So that's what CAM is actually used for. Though there are certain challenges. So policy, safety, you're not sure about the chiropractors, whether, for example, if a chiropractor tries to fix your slip disc, you don't know how it's going to be at the end and how many sessions you need for him or her to set it up. Then there's the efficacy, quality. You're not sure about the quality of the therapy, so you might even want to go to the modern therapies available. Availability, you're not too sure about how available these traditional medicine practitioners are. You'll have to look for them. Then preservation, research, and further development of these types of healthcare systems. So there are 2 different concepts behind the traditional medicine. There's the medication therapy. This utilizes herbs, animals, or animal parts, so it's biological based. And the non-medication therapy, this is where you involve in physical, mental, spiritual, and mind-body therapies. So basically yoga, meditation, and working out, basically those. Then there's the manipulative and body-based practices, energy medicine, and as I said, yoga therapy, and meditation. So I don't want to really dwell into these, But there's divination, spiritualism, and biological-based healing, as that's a repeat of what I've just done. Okay. So examples is shaman or Laban in the Kenyan system. Tanzanian is Niem Niem, and Africa again is Semgoma. Ghana is a Komfo. Okay, so basically these are just examples. There's the Twina from China. Okay, if you allow me. Okay, so basically what they do is they try to locate or pinpoint the imbalance in an individual's physical problem. The 6 eternal causes of disease, which is wind, cold, heat, dumbness, dryness, and heat. So there's 7 internal causes, which is joy, anger, sadness, pensiveness, grief, fear, and fright. I don't really need to go through this, so to just save some time. 5 elements: wood, fire, metal, earth, and water. So this is just the elements and the examples of the season, the taste, the emotions, and the body parts. Yes. So these are the common herbs. The Ephedra sinica, common name is the Chinese ephedra. Traditional use is anti-asthmatic. So pharmacologically active alkaloid, ephedrine, has been isolated, which is sympathomimetic alpha and beta adrenergic receptor agonist, causes vasoconstriction, bronchodilation, increased cardiac contractile activity, central nervous system stimulation, and weight loss. However, it's a potential drug that can be abused. So there's also the Artemisia annua. There's the Artemisia annua. So this is the artemisinin, where artemether is the methyl ether. Artesunate acid is succinic acid hemister. That is the sodium artesunate. Anti-malarial in combination therapy. It's not soluble in water. There's also the Artemisia. Yeah, you can actually confuse it with the A annua, Artemisia afra and A annua. So this ginseng. So ginseng is an anti-inflammatory, antioxidant, neuroprotective, cardioprotective, anti-diabetic, anti-cancer. It also increases the psychological and immune functions. So I feel like this is also a potential drug abuse because of the psychological effect. And then there's the phytochemicals, which is the triterpene, saponins, ginseноids. So so this is basically what I've actually talked about. It's just in detail, so allow me to just skip. So the Chinese have come up with something called acupuncture. If you go towards the Aga Khan Road in Six Parklands, you'll see there's a institution that provides acupuncture as acupuncture services. So here it alleviates nausea after surgery, chemotherapy, and pregnancy, used in treatment of headache, asthma, stroke, rehabilitation, and fibromyalgia. It can also be used for chronic pain. So there's the aromatherapy, where this is where you use essential oils. You mix it up in your water, put it in your bowl or anything, and then you can even use the scented candles or something, and then you just light it up, and you allow yourself to heal. And it's also used to combat insomnia, jet lag, pain, arthritis, rheumatism, stress-related illnesses like epilepsy, asthma, heart attacks, or heart conditions rather, and toxemia in pregnancy. So it's usually prepared in sunflower oil, and chamomile oil can also be used. So basic principle of acupuncture is, as I said, the previous one, they're just gonna use the concept of yin yang. So yin yang is it's a balance, so it needs to be balanced. So when there's an imbalance, there's the results to deficiency of excess chi. I think it's chi. I'm not a Chinese, so I wouldn't know how to pronounce. Okay, so let me just take you guys through the main things. So moxibustion, as I said, involves the burning of moxa wool, mugwort. Okay, so so this is what moxibustion is. It's like incense stinks, but sticks. Why do I keep saying that? But now in blobs, if I may say. Yeah. You just take it next to your skin and let the therapy begin. Do you guys have any questions?
No, not so far.
Okay. So tinwa means twi means push and na means grasp. So it involves soft tissue, spinal and joint manipulation. So here you'll use massage and okay, so it involves the manipulation of spinal joints and other body organs. It involves grasping, pressing, rolling, round rubbing, holding, twisting, rub rolling, pushing, kneading, rotating and shaking and wiping. So for those of you who've gone for massages, it's basically what this is, tinwa. Yeah. So the basic principle, it promotes and invigorates the flow of qi and blood. It expels, clear, dissipate, and dredge pathogen factors. It regulates the qi, which is the yin-yang balance and blood. Harmonize and balance the yin-yang, as I said. Nourishes, strength, and support. Improves and regulates the zangfu organs. And lubricates and joints. Lubricates the joints and facilitates movement and yeah, relaxes the channels. So the Indian medical system is Ayurveda, Siddha. Unani is supposed to be Japanese originally. Oops. It's supposed to be Japanese. And then yoga, there's the naturopathy and homeopathy. Yeah. Okay. So Ayurveda promotes life rather than to fight disease, so as to maintain harmony between a person and a nature in order to attain optimum health. So the basic principle is treatment is based on knowledge on both the disease and the patient. So air, which is air, water, space or ether, earth, and fire. This particular dosh, so there's a vata, which is regulates catabolism, electrolyte balance, elimination and metabolites. Efficiency is increased by dryness. There's the pitta, which is regulates metabolism, optic nerve, body temperature, hunger and thirst, increases in body temperature. There's the kapha, regulates anabolism, maintains joint lubrication affected by sweat and fatty foods. So this basically wants you to reduce your fat intake. The three malas, it's also the three malas. There's the feces, urine, and sweat. Okay, so here it's a fecal matter is not properly eliminated due to diarrhea, constipation, asthma, rheumatoid arthritis, urine, UTI, cystis, gastric pain, sweat, skin irritation, improper fluid balance. So there's the 8 disciplines of Ayurveda, IMED, psychological disorder, pediatric treatment, geriatrics, aphrodisiacs, and eugenics, surgical treatment, and otorhinolaryngology. ophthalmology, this whatever it is, and toxicological studies. Okay. Oops, sorry. Okay, so from here, Unani is actually Greek. Knowledge of the state of the human body in health and illness. So there's the Greco-Islamic medicine incorporated in the 12th century. So basically, it's used for 4 humors. There's the blood, phlegm, yellow bile, and black bile. I don't really know. Okay, so plants used to strengthen or improve your health. Tabiat means health. So Allium sativum, Nigella sativa, all these, sinensis, glabra, curcuma longa, all these, Zinga officinalis, which is ginger, aloe barbadense, aloe. I think this should be, cucumber from the name, not too sure. So Allium sativum is onions. This is, I don't know, sinensis. Okay, so yeah, you guys will just go through these images. So some of the regimental therapy. Okay, so some of the it involves leeches, where you place leeches on your skin. Venesection, purgation, emesis, enema, diuresis, expectoration, cupping. Cupping is where you place cups on your back or any part of your body. So it with pressure, it removes the toxins from the blood. There's a massage, exercise, hydration therapy, ointment and liniments, counter irritants, bathing, and fomentation. So this is what leeching looks like. You place the leeches on your affected areas and they suck out the toxins. So this is what cupping is. So dietotherapy, this is where you control your diet. So if you're diabetic, you reduce your sugar intake. If you're a heart patient, you reduce your your cholesterol, and so on and so forth. I don't want to repeat myself with homeopathy. Drug preps. So drug source, from what guys had said, there are multiple drug sources. So there is plants, minerals, animals, and biological materials. So when you prepare, you can use the maceration and filtration method in water and alcohol. Mixture sometimes up to 1 month. Stored at controlled conditions for up to 1 month. Potency is prepared by serial dilution, and solution is then shaken vigorously with impact. There's a specific website I'm going to show you. It's going to take us through the extraction method. So if you allow me, I can just go to it. Is that okay with everyone?
Yeah. That's okay. That's okay.
Yeah, because this is just basically, I went through this notes yesterday. He's basically just repeating these things. But if you guys want to really go through it, there's these specific plants that are used as folk medicines and animals. So let me just pinpoint them. So there's the bushmaster snake, used in circulatory problems and vascular complaints. I think it's the venom that's used. It's also used for menopausal and premenopausal symptoms like hot flashes, palpitation, headaches, heat, bleeding, fainting and depression, ovarian pain, slow to heal wounds, throat complaints, constipation and piles, depression and schizophrenia. And then there's also the cuttlefish, which is used for PMS and exhaustion. Then there's the cuttlefish, what this looks like. Then the tarantula or the wolf spider, used for mental restlessness, heart disease, ovarian disorders. The onions are used for burning sensation and neurological pain. Also in cough, the belladonna, that's the Atropa belladonna, from where atropine comes from. It previously was used to dilate pupils and staring. So now it's staring eyes. Flushing due to blood circulation, ailments where there's extreme sensitivity to light. Epicac, and that's Cephalis.
How do you pronounce that species word, Divya?
Please help me, guys.
I don't know. I'm asking.
I think it's Epicaquana, something. I don't. Okay. So it's used for nausea, with or without vomiting. So these are just the plants. I think you guys can go through them. I have sent the notes already. So there's also the mineral-based, where you use arsenic for anxiety and fear. There's the gold, which is used for depression and suicidal thoughts. Oversensitivity to noise, taste, and music. There's the calcium carbonate, which is slow development of bones and teeth, joint and bone pain. Then there's also mercury, which is ailments of throat and mouth, such as tonsillitis. Sulfur, skin conditions, mostly eczema. Digestive disorders, mental stresses such as lack of energy, and willpower, forgetfulness, irritability, and indecision. So, as I said, yoga can also be used. Hypnosis is used. So there's aromatherapy based on 3 notes: top notes, middle notes, and bass notes. Top notes have the high volatility for uplifting and invigorating effects like basil, thyme, and sage. Middle notes like chamomile, fennel, and lavender. Bass notes like the patchouli, and the lang, and the sandalwood. So basically, chiropractor, electrotherapy, pouching, osteopathy, and biofeedback. Let me take you to, the extraction now. Sorry, there'll be these ads. I don't know why SlideShare has these weird ads.
You can download the PowerPoint so that you don't get the ads.
Okay.
Yeah.
I'll have to sign up, blah, blah, blah. Let me just stop sharing 1 minute.
And you don't have to sign up. Just send me the link. There's another website where it downloads slides from SlideShare for free without signing up.
Okay, let me send you the link I'm using.
Okay.
No, not this one. There you go. I've sent you.
Okay, let me download it and send it to the group.
Let me also just
Meanwhile, you can give us a summary of this stuff.
Okay, so basically, folk medicine is basically what we've done. Homeopathy, there's the aromatherapy. Basically, you use plants, herbal based. You can also use the natural traditional medicine. I don't know if you guys have, of course, your parents, grandparents, and all have these particular remedies, like in the Indian household. If you have a flu and a cough, you can take warm milk with turmeric added to it. It tends to suppress the cough. There's also the dawa, as I know we all take it when we have flu and a sore throat, which is a concoction of ginger, honey, and a bit of lemon in it. Yeah, basically that's what traditional medicine is. Hypnosis is also one thing. It's basically using your it relaxes your mental state. It relaxes your mental state. So basically you're put into sleep, a kind of sleep state, which allows you to relax yourself. Yeah. Basically, yeah, that's the summary. If you guys have a question, you feel free to ask, please. Any questions?
Nah. Yeah.
No questions.
Okay.
No questions. Regarding this homeopathic remedies,
Homeopathic, yeah.
is there any evidence to support
Is
their
it
usage, or has it used? Is there evidence-based proof that they work in treatment of this condition?
honestly, I've not really gone through the evidences per se, but there are a couple of countries that actually use this. Yeah. They people actually use it for asthma. You can also use it for the hay fever. There is also the allergies also. So you see, what homeopathic actually means is that you use okay, you're treating your condition with okay, I don't know how to explain it. It's basically like with like. It's a treatment with like to like, and then it's where you stimulate the healing responses to diseases by mimicking the symptoms of those diseases in healthy people. So that's what I'm just reading right now. I'm not too sure if there's evidence of it that I'm not too sure whether there's evidence, use that people
Actually,
use
there isn't
effective.
any evidence that supports that it
Yeah.
works.
Yeah,
So
exactly.
I don't understand. Yeah, because personally, I'm trying to understand why, like, you would do 1,000 serial dilutions of a venom, and that
Mhm.
only would end up working.
Yeah, exactly. So it can also be toxic, but
Seems more
that's
like a placebo.
exactly. It's not really proven to be effective. Yeah.
Wait, so
Wait, but
Divya,
Uh-huh.
so homeopathy is essentially using this crude drugs.
Yeah, basically, yes, that. Yeah.
Okay. So
Yeah. Any other questions or we move? Okay, so medicinal plants. So this is the extraction bit I'm going to take you through. So medicinal plants are richest in their bioresources, modern drugs of traditional systems of medicines, modern medicines, nutraceuticals, food supplements, folk medicine, pharmaceutical intermediates, and chemical entities for synthetic drugs. So we'll go through phytochemicals more in depth later on, but phytochemicals are natural bioactive compounds found in different parts of the plant. There's a fruit, flower, stem, leaf, and root. They have the inhibitory effect on all types of microorganisms in vitro, widely used in human therapy, veterinary, agriculture, scientific research, and countless other areas. So extraction is basically a method used in separation of medicinal active portions of plant tissues using selective solvents through standard procedures. So the basic parameters influencing the quality of an extract include three of these factors. So there's the plant part used as starting materials, solvent used for extraction, and extraction procedure. So when you're choosing a solvent, you need to make sure that it's a good solvent and it's not toxic to the body. There is also the ease of evaporation at low heat, promotion of rapid physiological absorption of the extract in the body, and the preservative action. It should stay for a longer time, for a month or so. Okay, so the factors affecting the choice of solvent are quantity of phytochemicals to be extracted, the rate of extraction, the diversity of different inhibitory compounds extracted, ease of subsequent handling of extract. So something like ethanol, you need to make sure, since ethanol is flammable and also it can evaporate easily, you need to make sure you have proper handling techniques when you're dealing with ethanol. And the toxicity, you need to choose a solvent that is less toxic to the body, because remember, you're treating yourself, you're not toxicating, if I may say, yourself. Okay. So the most commonly used solvents for the active components are water for anthocyanins, starches, tannins, saponins, terpenoids, polypeptides, and lectins. There's the ethanol is commonly used for tannins, polyphenols, polyacetylens, flavonols, terpenoids, sterols, and alkaloids. So these methanol, chloroform, ether, and acetone are most commonly used solvents for these particular biological, rather active components. Yeah, so the techniques of medicinal plant extraction, there's the plant tissue homogenation. The explanations will come as we go. So this is just the list of them. Plant tissue homogenation, maceration, infusion, percolation, digestion, decoction, succinylate extraction, and sonication. If you guys remember what we did with Nelly, she had actually taken us through this very well, but I'm just going to give you a summary. So plant tissue is basically where you dry or you wet your fresh plant parts, and they're grinded in a blender to fine particles with certain quantity of any solvent. As I said, the plant that you use and the compound that you use, you're going to use your specific solvent according to them. Right? Like if you have a phenol, you're going to use acetone as your choice of solvent. Then you shake it vigorously for 5 to 10 minutes and leave it for 24 hours, which then the extract is filtered. The filtrate is basically what you're left in the funnel, the filter paper. So it's dried under reduced pressure and it's redissolved in the solvent or centrifuged for clarification of the extract. Maceration is where you use the coarsely, that is, there's a few thick particles in it. Powdered crude drug is placed in a stoppered container with the solvent, and you allow it to stand at room temperature for a period of at least 3 days with vigorous shaking until the soluble matter gets dissolved. Then the mixture is strained. The mark, which is the damp solid material, is pressed. The combined liquids are clarified by filtration or decantation after standing. Infusion is basically you heat your water and then you place your leaves or your bark or something, you boil it, and then you let it stay there. And then you can filter your, if you wish, you can filter the solution, and you can collect it in a container. Okay, so digestion is also a form of maceration in which gentle heat is used during the process of extraction. You use moderately elevated temperature, which is not objectionable. Remember, you do not want to denature. You do not want to denature, so you're gonna use it at a particular temperature. Solvent efficiency of the menstruum is thereby increased. So this is a microwave digestion system. Decoction is the crude drug is boiled in a specified volume of water, that is 1:4, for a defined time. The volume is then reduced to a fourth of the original, cooled and strained or filtered. So suitable for extracting water-soluble and heat-stable constituents typically used in Ayurvedic extract. So if you guys know neem, there's the mint, this is what you use. This method is what you'll use for getting the extracts. So percolation is used frequently to extract APIs in the preparation of fluid extracts. The solid ingredients are moistened with an appropriate amount of specified menstruum. Allowed to stand for approximately 4 hours in a well-closed container. After stand time, the mass is packed and the top of the percolator is closed. Mixture is allowed to macerate in the closed percolator for 24 hours. So I just want to explain what a menstruum is. It's basically a solvent used in the preparation of drug. It's a solid formulation of a drug. Okay. So the menstruum is added as required until the percolate measures about 3/4 of the required volume of the finished product. So the mark is basically what is in the filter paper, the wet residue is then pressed and the express liquid is added to the percolate. Sufficient menstruum is added to produce the required volume, and the mixed liquid is clarified by filtration or by standing, followed by decanting. So this is oxalate extraction, also known as the hot continuous extraction. Finely ground crude drug is placed in a porous bag or thimble, which is placed in the oxalate apparatus. It's basically heated and the vapors condense in a condenser. And since the condensed extract drips into the thimble containing the crude drug, then the level of the liquid in the chamber rises to the top of the siphon tube. The liquid contents of the chamber siphoning to the flask. Process is continuous and carried out until a drop of solvent from the siphon tube does not leave the residue when evaporated. Sonication is basically using the ultrasound to extract your APIs. So you can use 20 kilohertz to 2000 kilohertz, which increases the permeability of cell walls and produces cavitation. Okay, so there's disadvantage, that is the deleterious effect of ultrasound energy on the API, which forms the free radicals and undesirable changes in the drug molecules. So basically, you can't really use this in the body because it might have a negative impact. So the effect of okay, so that was your extraction method. So remember, there's
Divya,
yeah,
like could you go to slide? Yeah, that slide. Yeah.
Yeah.
Could you just kindly just summarize? Like, hey, you've gone fast there. Like just
Oh no, okay.
summarize the individual ones and what in basic terms, not even using those nini terms, in basic terms, what each of
Okay.
them in
So,
each does. Yeah.
okay, so basically plant tissue homogenization. Here you use mortar and pestle. So here what you do is just a minute. As I said, you blend and then you leave. Okay, so you're gonna add your solvent in the. Okay, so let's say you're using a mortar and pestle, right? You're going to grind or you're going to use a blender. You're going to add your solvent to it. You're going to let the extract stand. But again, you're going to shake it vigorously so you dissolve the thingy, the plant extract properly. You get. So here you're going to dissolve it. Do you guys get me? Hello?
Yeah.
Yeah? Okay. So that's basically what tissue homogenization means. It basically means that you're breaking down the tissue to get the extract. Okay, basically, all these techniques, you're going to break down the tissues and the cells in order for you to get the extract, yeah? So
Divya, go back to the homogenization.
Yeah, sure.
Okay.
Yeah, okay. So maceration is basically you having the coarse powder, yeah, and then you're going to dissolve it in a particular solvent like let's say ethanol, acetone, and you're going to leave it soaked for I think 3 days. That's what 3 days. You're going to leave it for 3 days, and when the 3 days have surpassed, you're going to collect you're going to basically filter it so that you get you guys when we used to filter, you remember the residue that used to stay in the filter paper?
Yeah.
You're going to press that to get the extract, and then you're going to again filter it again.
So that stuff that remains on the filter
Yeah.
paper, that's your mark.
Yeah, that's your mark.
Okay.
Yeah. And then the other one is infusion. It's just like tea leaves. When you're boiling water, you add your tea leaves. Yes. And if you want the black tea or black coffee for that matter, you're just going to strain it, and you're left with the black coffee and black water tea, that black liquid. Yes, that's infusion.
Okay.
You get? Yeah. So you're basically using water for infusion. And then for percolation, what you have to do is basically in percolation, the principle is that the water moves downwards through it moves downwards. So you're going to have let me just take you there. So here it's basically this. Can you guys see this?
Yeah.
So you're going to put in your extract, yes, and you're going to add in your solvent. It's usually water, yeah, and then you allow this container, the well that's here with the thimble here, you're going to let it stand for approximately 4 hours for the water to go through that plant extract. So you get this what do you call this? The solvent, the solution at the end, the extract. Do you guys get it?
Okay, so in this, let's say we are using let's say stem.
A stem,
So
yeah.
so like how would we go about it?
Okay, for
So
the
like
stem, you're gonna,
mm-hmm.
of course, you're gonna blend it a bit, yeah, to get this particular, this compound here.
Yeah, the smaller, yeah.
Yeah, to decrease the particle size. So there you're gonna add some water to it or any solvent of your choice. But again, you're gonna use the solvent according to the plant type, the compounds. Yeah, so then you're gonna leave this standing for approximately 4 hours. And to make sure that evaporation doesn't happen, you're gonna cover it. You're going to cover the thing. So you're going to let the water sip through that extract in order to form this this desired extract that you're going to use as your traditional.
Oh,
Yeah.
so let me annotate your screen a bit.
Yeah, no problem.
So for example, this is our grinded stem here, right? Then we put the water, for example. We put water here. Then somewhere here there is like a small hole that water is passing through here, extracting the ingredients, and then we collect it here, right? Okay. Now, okay, what is this mass now? The mass is that, now what's that?
After stand time, the mass is packed and the top of the percolator is closed. So you remember the stem that we blended?
Yeah.
That's your mass.
Oh, that's my mass.
Yeah. So you're going to
after,
leave it for 4 hours and you're going to put your water through it. Yeah. So
then the mixture is allowed to, okay.
Yeah. Your menstruum is what you're going to use as your solvent. Oops. Yeah.
So for example, in our example, our menstruum was water.
Water.
Okay.
Yeah,
So thanks.
You get it? Any other question, guys? Okay, how do I clear the annotation?
Let me see. Ama, you can
I
clear.
have a question. Is it just me, or are these things almost the same? I'm not getting like
Yeah,
the
they are the same.
differences. Okay.
What's the difference between maceration and percolation?
The difference between maceration and percolation. Okay, so you see, in maceration, there's not much of a difference, to be very honest. Yeah, so here, basically, for maceration, you're allowing it to stand for 3 days. In percolation, you're letting it stand for 4 hours. Then the mixture is then strained. So in percolation, you're just putting your mass in your thimble, your funnel, and you're adding your water to it, so it sips through it. But in maceration, what you have to do is, the mark that you stay with, you have to put it through a filter. You have to filter it again. It's like a double filtration for maceration, but one filtration for percolation. Are you getting it, Janet?
Kind of, yeah. So
Yeah.
for percolation, it's like if we go back to the previous one for percolation.
Yeah.
I think what
Yeah.
I'm getting
So
is
like
if it's in a question and they ask like if they specify the number of hours, if they say the process took 3 days, then definitely it's maceration, not percolation, because percolation is taking a shorter time. So perhaps to get the differences between these processes, you have to look maybe the time taken and the solvent used, because in the other one also Divya talked about there's what used specifically for maceration, because I'm seeing over here it's not there.
Okay, for maceration, you can use anything. It just depends on your plant.
Okay.
So if you have something like anthocyanins, remember you're going to use water. But if you have something like phenol, you're going to use acetone. It just depends on the biological compound of your plant. Do you guys get me?
Yeah, I think we can also say that, like when we're doing percolation, we usually use those, like the paper you said, so like a percolator compared to maceration, where I think we just let it stay still.
Yeah.
Yeah.
Okay, I've sent image for maceration versus percolation. Maybe you guys can go through it. It's basically what I've said, but now it's simplified, right? And
Yeah.
So decoction is basically so your crude drug, which you blended, is boiled in a volume of 1:4. So your water will be 1 and your crude drug will be 1, but your water will be 4. So 1 part of crude drug into 4 parts of water. Yeah. So you're going to heat it up to reduce the volume of the initial. So if it's 1:4, let's say it's going to be 50 mils. So you want it to reduce to approximately a fourth of 50 mils. Yeah, so you're going to end up with 12.5 mils of the original.
Yeah.
You get it, and then you cool it, and then you strain it, and then you obtain your extract. Yeah, so I think that's the end of the extraction methods. Soxylate. Okay, what? Let me just search what a soxylate apparatus looks like, because personally, I'm not even too sure about what a
I think it's in the next slide.
Is it?
Yeah.
Yeah, this.
Yeah.
So here, what you do is you're gonna heat it up, but then again, you're gonna put your mass, that is the blended or your powdered plant product, and then you're gonna add water to it, and then you're gonna cover the apparatus till no water gets out of the residue until the residue is literally dry. Evaporation, hot continuous heating. Do you guys get it?
Yeah.
Yeah. Okay.
Yeah, we do.
Ultrasound sonication is basically the use of ultrasound. So you're going to use a particular frequency, so ranging from 20 to 2000. And this increases the permeability of cell walls and produces cavitation. So through this, you're gonna, once the cell walls are permeable and it produces cavitation, it basically means that you're breaking down your cells to release the extract. But again, it's not recommended because it can cause a change in the plant extract by forming free radicals. And we do not want free radicals in our bodies. Yeah. Is that okay? Everyone okay?
Yeah. Thank you so much.
Yeah, so that's the end of extraction. I think we can go through now the secondary metabolites. These are just the tests for the qualitative analysis, which Willie will be taking us through at the end.
Nice.
Thank you, guys.
Welcome.
All right, someone else can now go through the secondary metabolites, or you guys want to do it some other time?
We take a break and then we continue.
Yeah, we can take we can start at 11:10.
Yeah, 6 minutes. Okay. All right.
Hey, guys. Hizo practical qualitative analysis hizo sinazifanya the end kabisa.
Tunazifanya?
Nitazifanya at the end, and then I was thinking maybe we can we can have like a longer timetable in terms of how when we do what, so that everyone can be well prepared.
Yeah.
So like for example, is you see just we're doing saponins, and the only way we can present saponins is if Stano has done like so that for saponins a type of glycosides. So
Yeah.
I think during the day we can stretch out over the week to do when we'll be doing what on Thursday. No.
I think we'll do that today. See from tujipange. Yeah, so that everyone can prepare even accordingly.
And also Divya, very great job. Thank you.
Yeah.
I was just saying you did a really good job. We are very
you.
grateful to have you.
Thank you.
So I'll see you guys at the 11th time.
Divya, is it the slides for biochem? Like that's what you've downloaded them, Ama, you'll be using SlideShare?
For biochem, I thought we were doing the secondary metabolites first before the biotech.
You've already sent the slides.
No, the slides were already on the group. I assume, I think it's what Sam had sent on the group.
Okay. Kindly just resend because I'm scrolling, I'm not finding them.
Okay, let me just highlight them.
Oh, highlight, yeah. Thanks.
Yeah. Or you guys want to go through plant biotech and genetics before we start? Because I'm seeing the secondary metabolites and all these things might take a lot of our. We will have to discuss it a bit in depth.
Kuna odaki.
Yeah.
Yeah.
So it's here.
Okay, sorry guys.
Okay, so plant biotech, application of scientific method to manipulate living cells or organisms for practical uses. So basically, he's the one who came up with it. There are two people. So there's the Matthias Schleiden and Theodor Schwann in the 19th century, and Gottlieb Haberlandt, German botanist, produced the plant tissue culture. So there are seven methods of plant biotech. So there's the gene transfer. So gene transfer is insertion of unrelated genetic information in the form of DNA into cells. This is called transgenic organism. Can be done in two different methods. So there's the vector-mediated gene transfer and the direct gene transfer. Here, you're going to use genetic engineering for gene transfer. So in the vector-mediated gene transfer is carried out by a factor called Agrobacterium, which is a mediate transformation. Agrobacterium tumefaciens is a soil-borne gram-negative bacterium. And there are two particular species of Agrobacterium. There's the tumefaciens, which induces the crown gall disease, and the rhizogene, which induces the hairy root disease. So it's a phytopathogen treated as the nature's most effective plant genetic engineer. So this is how the gene transfer is done. So the plasmid is removed from the bacterium, and the tDNA is cut by a restriction enzyme. So the bacteria has the plasmid, which we will, of course, remove it. And using the restriction cleavage site, you're going to cut the tDNA, where a foreign DNA is cut by the same restriction enzyme, right? And then okay, you can see the colors. This is gray and this is yellow. The yellow one is a foreign. So you're going to take the foreign DNA is from something else, another plant per se, or plant. So the foreign DNA is then inserted into the tDNA of the plasmid. Okay, just a minute, guys.
Take your time, take your time.
Okay, as she has taken a minute, yeah. Let me ask you guys something. How have we decided with the scraps issue? Because I'm seeing time is really going. If there are people who are willing to get Jambi scrubs, we can come together and then we can buy. Also, if someone else is willing to buy elsewhere, it's also fine, because now I'm seeing like time is really moving.
Amesema hauzi anymore.
Oh, Njambi hauzi.
Yeah. But I've asked if she
So
can
what?
plug us with her supplier, but we text the supplier ourselves. Alternative, letting ako hapa kwa hii meeting.
Letting ako hapa?
Yeah. Let me see. I don't think she has joined.
Anyway, pia kuna letting alikuwa anasema Dr. Nas. We can also get from Dr.
Me,
Nas.
I don't want Dr. Okay, I can get Dr. Nas. Dr. Nas are the ones they have to get your measurements and fit you. Yani, ukinunua Dr. Nas, you better not put on weight. That is the catch. It's fitted.
Done for that.
Between now and 13th of next month, how much weight can you gain? I doubt if anyone is capable of gaining so much weight within the time that we have between now and then. Kumbuka pia, tukimpatia measurements kama maybe between today and tomorrow, time atachukua maybe by the end of this month ndo zitakuwa ready. So I don't think zitakuwa mbaya pia. But yeah.
All right. Thing is, people don't talk, people don't communicate. I think, I don't know, it is what it is, but me, I'm thinking nikipata hiyo supplier wa Njambi, mi nitamuongelesha kivyangu, hata kama nitauda pea moja, mi nitauda yangu. Yeah.
Yes, huyo Njambi pia, like the name will be there, like hiyo price yake ilikuwa plus the name tag.
Yeah, the price she was giving us was for everything. You get the scrubs, you get the lab coat, and both of them are branded.
Okay.
Ukaona kwa group watu wakaanza yeo, now finding out everything is more expensive. Yeah. But it is what it is. Mambo tu huwa ni mengi. Let us go back to the discussion.
Sorry, guys. So basically, you're going to remove the plasmid from the bacteria and the DNA from a plant. So here you're going to insert the tDNA of the plasmid into this the remaining plasmid, and then the plasmid is then reinserted into the bacterium, where the bacterium is then used to insert the tDNA carrying the foreign gene into a chromosome of a plant cell, where then the plant is then grown in culture, and then shifted to the soil where a clone is formed of these plants. So basically you're using the GMO, the technique of genetics to form the GMO plants. Do you guys get it?
Yeah.
No, me, I've not got it, to be honest. Abu, say it again.
Okay, so you okay, so this is your bacteria. Yes, you're gonna get your plasmid, since the plasmid is what is the circular it's the genetic info, it holds the genetic information, right? So you're gonna cleave it and you're gonna cut a particular point in the plasmid. You're gonna cut a particular point in the plasmid to insert another piece. Where you're going to cut, you're going to insert another foreign piece into the plasmid to form a full plasmid, which is then inserted back into the bacteria. Yes? Do you get it, Saru? Are you following?
Yeah, I'm following.
Yeah? Okay, so you're going to put it back into the plasmid, where since the plasmid is the one that contains the genetic information, is going to form its chromosome, right, with the foreign and the plasmid. So the foreign gene and the plasmid form a chromosome. This chromosome is then extracted and the bacterium is then cultured on a plate. Basically what we do in labs, it's cultured, and then once the plant is grown, you transfer it to a soil and you let it grow. So whatever is growing is the clone, and that's how the GMO plants are produced. Are you getting it?
Yeah.
Yeah? Okay. So plasmids versus as vectors. So a vector is an agent that carries the gene from one organism to another. And plasmids are self-replicating circular DNA molecules in bacteria, as we said. So vectors which carry gene in plants are the tobacco mosaic virus and the cauliflower mosaic virus. So, okay, Gaius,
Yeah.
do you know how to explain the tobacco mosaic virus and the cauliflower mosaic virus? Because personally, I don't.
Would you have their place they've written it down, they've just thrown
No,
a
they haven't.
there.
But I feel like it's the same. It's the same principle as this.
Let me.
If any other person knows, please feel free to explain.
To explain what? The process you've explained.
The vectors, the tobacco mosaic vector and cauliflower mosaic virus.
Let me see. To scroll to the
next here.
Oh, nah.
I'm seeing is essentially the same thing. If we go back to the previous slide, that one. It's the same thing, only now you're using a virus to deliver the altered DNA.
Yeah.
Because viruses will enter the cell and deliver their genetic material. So it's the same thing we use even in humans.
Okay, I've just gotten a image. So here it says inside the protein capsid, there's a single-stranded RNA molecule, which is spirally coiled. Is linked, this protein subunit is linked to 3 nucleotides of RNA, and in an arrangement of capsome of RNA. That's all I have. So there's not really I don't know. What I'll do is I'll find a better explanation and I'll send it. I'm so sorry, I'm not able to explain this right now, but when I get a better image, I'll send it to the group with a small explanation for both of these. Okay, so restriction enzymes and DNA ligase, when Gaius was taking us through this, restriction enzyme and DNA ligase are used to make the recombinant DNA. So the restriction enzyme is a DNA cutting enzyme that recognizes specific site in DNA, and the DNA ligase is a glue. It acts as a glue. It joins the two strands together. Yeah. So the steps involved are DNA fragment is firstly isolated, and then the restriction enzyme will cut off the DNA, and the DNA ligase will then join the DNA. Yeah, the target gene is what will be cut by the restriction enzyme. So cloning is the process of genetically producing an exact copy of a gene, cell, or organism.
Divya, can you go back to the, sorry, can you, this, yeah.
This, okay. You want me to explain?
So yes, please, again, kindly.
Okay, so you've chosen your particular species, yes. You've extracted your DNA that you desire. Yeah. So in that DNA, we all know DNA is made up of genes and all. So when you're cutting your DNA, you're gonna target your DNA. You're gonna target a particular gene. So let's say you're gonna target your insulin gene, right? Which is gonna be cut by the restriction enzyme. Yeah. So this is your plasmid. So this target gene is cut by your restriction enzyme, and you're going to join a foreign gene. You're going to join a foreign gene by DNA ligase to form a recombinant plasmid.
Thank you.
You're welcome. So cloning is making multiple copies of the recombinant DNA. A clone is an exact copy of a gene, cell, or organism. It involves modifying pre-existing genes to form new genes, and the most common plasmids used are PBR322 and PUC18. I don't know whether we really need to know this. Okay, so there are 4 steps involved in cloning. There's the isolation, ligation, transfection, and selection. So let me just, so you're isolating a particular gene that you want. Let me just get you a proper, what do you call that? A proper, a picture for it. Gene, let me just send it to this group with the, yeah, here. So that's your steps for DNA cloning. So if you guys go to your WhatsApp, I've sent an image for it. Yeah, let me just try projecting it here as well.
Yes, we can see the image, Divya.
Yeah, you can. Okay, so
yeah.
Okay, so isolation basically means you're isolating your target gene that you require to form your recombinant DNA. Ligation, okay, so here, according to this image, so you're going to clone your vector, which is the plasmid, in which you're going to cut off your target gene using the restriction enzyme. So from the eukaryotic chromosome, you're going to select a DNA fragment, which you target gene from, rather the target DNA fragment that you want to clone. And again, you cut it using the restriction enzyme, so restriction endonuclease. So the plasmid and the particular gene that you've taken from the chromosome are joined together by DNA ligase, where a recombinant vector DNA is introduced into the host cell. Yeah. And then propagation happens, which produces many copies of recombinant DNA. You guys are okay? Or you want a detailed explanation?
Personally, I'm okay.
Okay, yeah, all right. So this is basically what I've just explained using the simplified diagram. If you guys can go through it at your own time. The notes are already on the group. Multiple copies are formed here after the DNA propagation. Yeah, okay. So the polymerase chain reaction in clones DNA without using cells. PCR takes place in a small tube that contains a sample DNA called primers. It includes all 4 nucleotides of coding. So there is the adenine, thymine, guanine, and cytosine. Yeah. Can you guys even see this? Okay. PCR. Let me just get a diagram for it and see. Guys, do you know how to explain the PCR, or anyone else for that matter? Personally, I don't.
Chai knows. Chai, we were talking about it the other day. Maybe he can.
Okay, so I've just sent a image, the last image I've just sent. Maybe we can use that to explain. So polymerase chain reaction is, you use the DNA template where you denature by heating at a minimum of 94 degrees. The heat then breaks the hydrogen bonds between these nucleotides and separates into single strands. And then you anneal, whereby you use the DNA primers. DNA is then cooled down to 50 to 60 degrees, where the DNA primers bind to the individual single strands. And this causes extension by DNA polymerase, which inserts nucleotides and extends the new strand. I don't know if that made sense to you guys.
Yeah, so essentially, it's just you're synthesizing a new a DNA strand in vitro. That's what.
Yeah.
Yeah.
Yeah. So there are 4 ways of inserting gene clones into plant cells. There's a gene gun, electroporation, micro-injection, and liposome. So the gene gun is a biolistic particle delivering system originally designed for plant transformation. So you're gonna target a particular part on particular plant cell that you want, and this gene gun barrel has a plastic disc with DNA coated gold particles, where these DNA coated gold particles go into the target plant cell. There's one thing here that I found, it's a better explanation. it's a better explanation. So you're basically bombarding the target cell with nucleic acid coated gold particles at a high velocity. So that's how you're going to clone your plants. If someone has a better explanation, kindly, because biotech is not my strong hold here.
Yeah, it's just what you say. It's a gun with the gene you're delivering, the altered gene that you're delivering to the plant. So you just pull the trigger and the gene is delivered. But now with the gene gun, because essentially think of this, let me annotate. Yeah, think of this as your leaf, and then you are having your gun here. Yeah, right?
Yeah.
So you shoot and it goes and enters the leaf. But it's with the gene gun, the method does not always work because you're just shooting it through the membrane to go to the leaf, but you're not guaranteed as to where it will go. You're not guaranteed it will actually go to the DNA. It may even go to the mitochondria. And another disadvantage, it may destroy the cell because it involves breaking this barrier when it enters. But it's what you just said. Gene gun, you have your gene here and you shoot. It goes so fast that it penetrates the membrane and enters into the cell. Yeah.
Yeah. Thank you. Then electroporation, you basically use a high electric field. You apply it to the cells to increase the probability of the cell. I think it's supposed to be permeability of the cell membrane. So
Yeah,
basically, yeah. So you're gonna the cell then takes up the DNA and gene clones are formed. So the advantage of electroporation is that it's simple and convenient. It's also, the cells are then transformed at the physiological state, at the normal physiological state. However, the disadvantage is the delivery is low for DNA and it has a low regeneration. So basically the plant clones will be slow. The plant clone production will be slow. Then microinjection, using of a micropipette to inject DNA into a cell. So this is just targeting a particular, you're just inserting an injection with the DNA into a plant cell, which then clones. Yeah, then liposomes are small spheres made of liquid molecules that fuse easily with plasma membrane. I'm not too sure about liposome. So it's okay. So it's absorption of cell membrane, basically. If someone has a better explanation, please feel free. From what?
Okay, with a lysosome, it's essentially we'll have the exterior and the interior. The interior is where you have what you want delivered, the X gene, for example. Then the exterior, now you want the exterior to mimic the environment in which you're in. So for example, because it's the same principles in humans as in plants. So let's say we wanted to deliver the X gene, let's say it's the gene to cure sickle cell disease, right, to a sickle cell patient. And we know that the cell membrane is a phospholipid bilayer. So that tells us it is hydrophobic. So we'll make the external will be hydrophobic here. so that when it comes into contact here, as shown here, the hydrophobic cell membrane is able to be like, this is our friend. So it can take it up, like it can easily, it's like it diffuses part of the wall of the liposome will diffuse here. So it will merge, then we'll be able to release the X gene to enter the cell. So in a liposome, we'll have a hydrophobic external and a hydrophilic internal. But it can also change depending on the environment. You can also switch them to be the opposite. The outside can also be hydrophilic, and then the inside be hydrophobic. Because liposomes, we'll also see this principle being applied in emulsification of fats, how bile works. How bile works is essentially how liposomes work. Yeah.
Okay, thank you. Okay, so there are two methods of tissue culture. There's the anther and the meristem. The anther culture means plant regeneration from a haploid microspore cells with the aim of haploid and dihaploid plant production. So you're going to take your anther, you're going to culture them in a nutrient medium. It forms a ambroid, which will give a haploid plantate, and also a callus, which gives you the haploid plantate. Then there's also the meristem, where the potted plant, you take the meristem and place it in a culture medium where new shoots are formed in the tissue culture, and the rooting and conditioning of transplanted shoots happens in a container, which then you transfer the hardened plants to soil or box containing nutrients. So genetic engineering, as we said, is transfer of genes from variety of organisms into plants. So from bacteria, you usually use bacteria. You can also use a virus as your vector. So these are GMOs. Basically, that's the end of what I'm to teach. Yeah, this is just extra information. So advantages of GMOs: nutritional content can be improved. Genetically modified foods can have a longer shelf life. Creates food that are more appealing to eat and easier to transport. Disadvantages are that can cause antibiotic resistance. Genes go into different plant species and independent research is not allowed. Yeah. So this is a GMO. It has more color compared to the normal, and it looks more appealing to the eye. Yeah. Golden rice and the normal rice. So yeah, that's basically that's the end. Thank you for listening. I'm so sorry I didn't have particular explanations for genetic engineering. Forgive me for that, but I hope you guys got it.
Yes,
I
yes,
have a
we
question, Divya.
Yeah.
Jasmila, go first.
Oh, no. You go first. Me didn't have a question. I just understood very well. Thank you, Divya.
So my question is, are GMOs good or bad? Now that you've taught us about GMOs.
Personally, I'm not up for GMOs because, as we say, they just add the nutrients, but again, it's also causing particular changes in your body. And also, as from what we've seen in the slides, it also causes antibiotic resistance. So now, for example, when you fall sick and you need the antibiotics, it may or it may not work. So yeah.
Okay.
Personally, I'm not for GMOs.
You're not for GMOs.
Yeah.
But the thing of antibiotic resistance, doesn't it involve perhaps using of strains that are already resistant? So can that disadvantage be ruled out with careful analysis of, for example, if it's E. coli you're using?
Yeah, of course it can be ruled out. Yes. Yes, and GMOs are usually more nutritious since, as I said, they add more nutrients to your plants. So like, yeah.
But now
It's
the question of it's changing your body, I thought it's changing the plant. The plant is being made more, then you are eating the starch and the likes. Like I'm not saying I support GMOs. I'm saying the arguments that are out there. Maybe Stano can pitch in. I know he's a big advocate.
You had this argument with someone in Urban Allied?
Even me. Yeah, that's the intern.
Ian. It's okay.
Okay.
I think GMOs are okay, but personally I wouldn't take them.
How do you know you haven't taken them? Now that's the question.
You've taken them, Divya.
I have? Okay, maybe yes. Maybe no.
You see, even these mangoes that actually come in seasons, most of them are grafted, and grafting involves some aspect of, I don't know.
Oh, okay.
Like, you go to the